Welcome to The Explainer. Today we're unraveling a literal life-or-death mystery happening right now, inside your head. Imagine there's a master switch inside your cells, one that holds the ultimate power of life and death. Well, we're going to look at some incredible new research that reveals exactly why our most vital cells, our neurons, sometimes suddenly and tragically decide to destroy themselves during disease or extreme stress. It's a fascinating story full of cellular detectives, molecular alarms, and a newly discovered mastermind.
Section 1. The Neuron Death Mystery. Let's step right into the molecular crime scene, shall we? What we're looking at here is a beautiful, healthy human neuron. Its complex, sprawling branches are what allow our nervous system to send messages, form memories, coordinate movements. I mean, they literally make us who we are. And because they're so highly specialized and really don't easily regenerate, keeping these cells healthy is absolutely crucial for our survival.
But tragically, under certain conditions, things go horribly wrong. In neurodegenerative diseases like Alzheimer's or Parkinson's, or even during intense cellular stress from treatments like chemotherapy, these vital cells get a fatal signal. And here's the crazy part. They don't just fade away randomly from wear and tear. Instead, they get a very specific order to essentially destroy themselves.
In biology, we actually have a specific word for this: apoptosis. It's the process of programmed cell death, where a cell effectively triggers its own destruction. So the absolute key takeaway here is that we aren't dealing with accidental death. This isn't a neuron just running out of gas. This is a highly coordinated, intentional inside job.
Section 2. Searching the human genome.
To solve a complex inside job, you need detectives. The researchers knew the death signal was coming from inside the house, right? But here's the problem. We have roughly 20,000 genes in the human genome. Out of all of those, which specific gene is the one that actually pulls the trigger when a neuron gets stressed?
To figure that out, scientists turned to a seriously brilliant tool called a whole genome CRISPR screen. Let me break down how this actually works. They took thousands of human neurons in the lab. Then, using CRISPR technology, they systematically turned off their genes, literally one by one. After that, they blasted all the cells with a toxic stressor, in this case, a chemotherapy drug called vincristine. Finally, they just watched to see which neurons survived. The logic is so elegant, if turning off a specific gene saved the stress cell from dying, boom, the researchers knew they had just found a culprit in the death pathway.
Section 3. Meet the master switch.
The results of that massive genetic dragnet weeded out thousands of innocent bystanders and pinpointed our prime suspect, ATF2. This is a transcription factor, which basically means it's a protein whose entire job is to bind to DNA and control how other genes are read. Now, ATF2 was previously kind of understudied in neuroscience, but this whole genome screen thrust it right into the spotlight, identifying it as the central driver and master switch of neuronal death.
Okay, let's dive into this. Because ATF2 doesn't just act on its own out of nowhere. Before it can issue that final death order, the cell first needs to realize it's actually in danger. That's where these two highly conserved proteins come in, DLK and LZK. You can think of them as the cellular alarm bells. They sit in the neuron, sensing stress, and when they detect damage, they kick off the deadly chain reaction that eventually reaches ATF2.
Section 4. The Chain of Command.
So, how exactly does the warning signal get from those alarm bells all the way down to the master switch? Well, it all starts with sensing the stress. An initial physical injury, like an optic nerve crush or toxic stress from a drug, disrupts the neuron. This immediate physical or chemical trauma is what initially sets off those cellular alarm bells we just talked about, DLK and LZK.
Now, to pass that warning signal down the chain, the cell uses a microscopic communication system called phosphorylation. As defined here, this is the process of adding a tiny phosphorus molecule, which we represent with a P, to a protein. You can think of this exactly like flipping a physical on switch. When a protein gets phosphorylated, it wakes up and becomes completely active.
So once the DLK and LZK alarm bells ring, they actively pass the danger signal to the next protein in line, a molecule called JNK. Notice those crucial yellow P on switches being added to JNK's structure right there, waking it up to carry the message forward. And here is where it connects to our mastermind. The newly activated JNK travels down the line and flips the switch on ATF2. You can literally see those P molecules snapping into place on ATF2. Our master switch is now fully activated, armed, and completely ready to enter the nucleus of the cell to do its ultimate damage.
Section 5. Activating the death genes.
Once ATF2 enters the nucleus, things happen really fast. Let's look at this like a molecular flipbook to see exactly how it executes its fatal order. First, our fully activated phosphorylated ATF2 protein zeroes in and binds directly to a specific target sequence on the neuron's DNA strand.
Now, what's really interesting here is that ATF2 doesn't work alone at the finish line. Look at what happens next. Another key protein, called c-Jun, arrives on the scene to act as molecular backup, forming a deadly complex with ATF2.
These two transcription factors, ATF2 and c-Jun, are now perfectly positioned together right on the DNA. They lock into place side by side. Honestly, it's very much like two keys turning simultaneously in a launch console.
And with the team fully assembled, the final order is executed. Together, ATF2 and c-Jun drive the transcription of the neuron's death warrant. They force the cell to manufacture the very proteins that will systematically dismantle the neuron from the inside out. The apoptotic process completes, and the neuron dies.
Section 6. Why this discovery matters.
Let's take a step back and reflect for a second, because we aren't just solving a molecular puzzle here just for the fun of it. This mechanism of cell death is highly conserved across mammals. That means the exact same master switch we see in human cells also operates in animals like mice. And by deeply understanding how this chain of command works in mouse models, researchers have a reliable testing ground to eventually apply these exact lessons directly to life-saving human therapies.
The ultimate takeaway from all this research is absolutely monumental. Targeting the master switch could save neurons from disease and toxic stress. By mapping out this specific chain of command, scientists now have distinct, visible targets. If researchers can develop new pharmacological drugs that block these specific interactions, say preventing J&K from phosphorylating ATF2 or stopping ATF2 from binding with c-Jun, we might actually be able to intercept the death signal and keep those neurons alive. We've uncovered the inside job, and we've identified the mastermind. So I want to leave you with this final, provocative thought. If we can finally develop the medical tools to turn off this master switch for neuron death, what currently incurable neurodegenerative diseases might we conquer next? Thanks for joining me on this explainer, and keep questioning the incredible science happening inside all of us.